Welcome to the Deep Dive, where we take complex topics, cut through the noise, and give you the essential insights you need to be truly well informed. Today, we're plunging into a world that's becoming increasingly relevant to all of us, the vast, rapidly expanding universe of biomedical data. Think about your own health records, right? Or the incredible scientific discoveries happening daily. This data is really the engine of modern medicine. But, and this is the focus today, it comes with some surprising hidden costs and challenges. Our mission today is to unpack this intricate relationship between privacy, the sheer explosion of health information, and, well, the surprising bottlenecks researchers are facing as they try to make sense of it all. We'll be drawing from a really fascinating stack of sources. We've got a deep dive into biomedical data privacy from PMC, studies on research data management from the Journal of the American Medical Informatics Association, recent insights on genomic data analysis from Nature Reviews Genetics, and a look at patient and public views on data sharing from the Journal of Medical Ethics, plus a recent report on scaling biomedical databases. Quite a mix. So if you've ever wondered how your health data is handled, or what it really takes to power the next big medical breakthrough, you're definitely in for a treat. Okay, let's dive in. So when we talk about biomedical data, privacy is usually the first thing that pops into mind. And it's not new, right? It's been around since the ancient Greeks even, and definitely reinforced with the rise of electronic medical records decades ago. But here's where it gets really interesting. The arrival of ultra-cheap data collection and processing tech is fundamentally changing everything.
It really is. What's truly striking here is how the traditional boundaries of healthcare are just dissolving. We're seeing health information collected everywhere now. Mobile devices, obviously, sensors in your own home, even things called body area networks, tiny sensors you wear. And at the same time, the sheer detail and the diversity of information are increasing at an unprecedented rate. You've got your standard clinical data, but now it's complemented by all this omics data like genomics, proteomics, and it paints an incredibly detailed picture.
And with this data explosion comes what our sources call a privacy invasion, which sounds pretty serious. We've seen these troubling cases, right? Healthcare providers snooping on records of famous people or even family and friends.
Yeah, that happens.
And personal info being used for identity fraud. Millions of records exposed because someone lost an unencrypted laptop or phone.
Right. So the natural next question becomes, what's the real-world impact of these breaches? It's not just theoretical. The sources reveal a pretty concerning trend. Patient trust is eroding. I mean, think about this. Between 15% and 17% of U.S. adults have actively changed their behavior to protect their health info.
15 to 17%, that's a lot.
It is. And it's not just worry. It means people are actually going to another doctor, maybe paying out of pocket to avoid insurance records. Sometimes not seeking care at all to avoid an employer finding out, giving inaccurate information, or even self-treating instead of seeing a provider.
Wow.
And specific vulnerable groups, adolescents, people with HIV, mental health patients, victims of domestic violence, they report similar behaviors. There was one survey, for instance, 15% of Californians said they avoided discussing depression with their primary doctor because of privacy concerns.
That's staggering. It really drives home how deeply personal this data is.
Exactly. The core insight here is that privacy isn't just some abstract legal thing. It's directly impacting public health behavior.
And our sources explain that privacy itself is, well, complex. It's multidimensional. It covers anonymity, confidentiality, solitude, and it's highly contextual, right? Your expectation of privacy is totally different talking to your doctor versus, say, a stranger on the bus.
Absolutely. And even how sensitive you feel specific health information is varies hugely from person to person. To manage this complexity, the journey of biomedical information can be thought of in 3 distinct zones. That's how the sources frame it.
Okay, 3 zones. What are they?
So first is the collection zone. This is where data is first gathered. Could be at a clinic, could be via a mobile app. Here, privacy is about who can collect it, how much, when, and for what purpose. It's about anonymity, limiting content, getting consent.
Got it. Collection zone. What's next?
Second is the primary use zone. Once collected, data lives in systems controlled by whoever's providing the primary service, like direct patient care or a specific research project. Here, privacy focuses on confidentiality and security. Who gets access? For what? How is it protected?
Okay, primary use. Makes sense. And the third?
The third is the secondary use zone. This is when data gets used for purposes different from why it was originally collected. Think repurposing clinical data for a totally new research study, or sharing public data sets for policy evaluation.
Okay, so the data takes on a new life, potentially.
Exactly. And this zone brings up unique issues of anonymity and consent, both for individuals and potentially for groups, because the original purpose has shifted.
That framework collection, primary, secondary use, that actually helps clarify things a lot. But how do we actually, address the challenges in each zone?
Well, the sources highlight two interacting tracks. There's a socio-legal track defining rights, responsibilities, policies, and then there's the technical track building controls directly into the information systems.
Okay, so law and tech working together. Can you give some examples?
Sure. On the socio-legal side, you see things like patients wanting really granular control over their electronic medical records. There are studies looking at how teenagers perceive health privacy on social media, which is a whole new can of worms.
Oh, I bet.
There's the privacy by design framework used in British Columbia for their population data. The U.S. HIPAA de-identification strategy is another example.
Yeah.
There's even a Supreme Court case, Sorrell versus IMS Health, all about selling prescription records. So big legal battles.
Right. And on the technical side.
Technically, HIPAA's security rules specifies safeguards like authorization controls for who can access EMRs. Though, interestingly, encryption of stored health data is technically addressable, not strictly required by HIPAA, which surprises some
Oh, it's
people.
not required, huh?
Yeah, and a study found that security practices really vary among U.S. healthcare organizations. There's also a significant insider threat. People who should have access may be looking where they shouldn't.
Ah, the snooping thing again.
Exactly. So data-driven auditing strategies are being proposed to track who accesses what. We also see de-identified data used a lot for research, like correlating web searches with actual visits to healthcare facilities. And there's even more advanced stuff, like private record linkage, matching records without a central identifier, and deriving secure genomic fingerprints to track patient records anonymously.
So this means really sophisticated methods are being developed, like automatically stripping out names and addresses from text using AI.
Yep, using both rule-based systems and machine learning. And for structured data, like lab results, there are techniques like clinically informed perturbation.
Perturbation, like adding noise.
Exactly. Adding a tiny bit of calculated random noise to, say, lab test results to make it harder to re-identify someone without really affecting the overall research value. Or the K-anonymity principle.
Right, you mentioned that. Where every record looks like at least K other records.
Precisely. So if K is 5, your data, even if unique, is indistinguishable from at least 4 other people in the dataset based on certain key identifiers, makes finding you much harder.
These technical fixes sound pretty complex.
They are, and if we look bigger picture, even more recent theoretical models are emerging, like differential privacy.
Differential privacy. Okay, how does that work?
Imagine you ask a question about a huge database, like what's the average age of patients with condition X? The system adds a tiny mathematically controlled amount of noise or randomness to the answer it gives back.
Okay.
Why? That noise cleverly ensures that even with the answer, you can't definitively tell if any single individual's information was included in that calculation or not. It protects individual privacy while still letting researchers get valuable aggregate insights.
Huh. That sounds incredibly powerful for balancing utility and privacy. But are there downsides? Does it always work perfectly, or does it, I don't know, make the results less accurate?
That's a great question. It offers strong mathematical guarantees of privacy, which is fantastic. But designing and implementing it effectively can be quite complex. And yes, there's sometimes a trade-off. You might sacrifice a tiny bit of precision in the aggregate results to gain that privacy. Making it truly practical and easy to use across the board is still an active area of research.
Got it. So, powerful idea. Still working out the kinks in practice. Okay, shifting gears a bit. What do everyday people, patients and the public, actually feel about sharing their health data for research?
Our sources from the Journal of Medical Ethics looked right at that. And generally, people express a strong willingness to share their data.
Really? Despite the privacy concerns?
Yes, primarily because they want to contribute to advancements in healthcare. There's hope for future personal health benefits, too, and even just pure altruism, especially among older citizens. They want to help the greater good.
That's encouraging. What benefits do they see?
They see significant potential. Better patient care through improved diagnosis and treatments, maybe more efficient use of healthcare resources, the ability to study long-term treatment effects or rare diseases, which you can only do with large populations. Some even believe it can spur innovation and cut down on waste in research.
But there's always a but.
Right. That willingness definitely decreases when people have higher privacy concerns or if they distrust government oversight, for example. Which brings us back to that critical point. How do you build and maintain trust, especially when breaches happen?
So who do people think should be responsible for managing this data sharing?
Ultimately, they tend to place the responsibility on researchers themselves. They suggest things like data access committees. These committees ideally would include maybe a researcher from the original project, a clinician, importantly, a patient representative, and maybe even a study participant.
So diverse perspectives making the decisions.
Exactly. And they also really value strong sanctions for data misuse. Accountability is key to trust.
Okay, so we've looked at the individual's perspective, the privacy paradox. Now let's pivot to the researchers themselves. You'd think with all this data flooding in, all this advanced tech we just talked about, labs would be humming along like well-oiled machines, right?
You would think so. But here's where it gets really interesting again. A study from the Journal of the American Medical Informatics Association paints a very different picture. It turns out that these rapid advances in analytical technology, combined with widespread access to large, complex biomedical data, have actually dramatically increased the difficulties researchers face
Increased. How?
in acquiring the data, archiving it properly, annotating it so it makes sense later, and actually analyzing it. Investigators are finding data handling itself is a major barrier. The computational solutions they need are often way too expensive or too complex to implement, or sometimes they just don't exist yet for their specific dynamic needs, which can literally change week to week.
So given these huge data handling problems, how are these cutting-edge scientists actually managing day-to-day? Are they using super sophisticated custom tools?
Well, this is the surprising part. The study found a widespread use of non-specialized applications for core data management,
Non-specialized. Like what?
like general-purpose tools, spreadsheets, text files, simple file sharing programs. We're talking Microsoft Excel,
Yeah,
basic.
kidding. Spreadsheets
Yeah.
for biomedical research data.
I'm not kidding. One researcher in the study literally said, quote, "Yeah, the spreadsheet has been our main workhorse, unfortunately." Another shared, quote, "I have one spreadsheet that has all of my chromosomes. It has all of my data. It has my whole experiment."
Wow. Just, wow. That's genuinely astonishing.
It really is. The startling takeaway is that despite all the cutting-edge science, a significant chunk of biomedical research is bottlenecked because scientists are managing like terabytes of complex data using Excel. It's like building a rocket with, I don't know, hand tools.
Okay, so they use them because they're simple and available, I guess.
Mhm.
But what are the real downsides in a research context? They must know it's not ideal.
Oh, they know. 9 out of 15 researchers interviewed recognized the disadvantages loud and clear. Size constraints, for one. Spreadsheets just bog down or crash past a certain point, maybe 20,000 cells, the study mentioned. Limited processing power. They're cumbersome for printing or trying to link data between different files.
So they know these aren't the right tools, but they're stuck using them.
Pretty much. Out of necessity, it seems. And this reliance on ad hoc methods leads straight to another major problem. Just organizing, storing, and retrieving the data becomes a nightmare.
I can imagine.
Researchers are dealing with incredibly complex, varied data formats. The number of files and their sizes just keep growing, and indexing and annotating consistently, forget about it sometimes. Many just prefer their own individual, often informal methods. Some admitted to having no organizational methodology at all.
No organization at all. That sounds chaotic.
Picture this quote. They're not organized in any way. They're just thrown into files under different projects.
Oh, my goodness.
And you've got separate files on different computers that don't talk to each other, so finding something specific later, good luck. Many aren't even aware of basic search tools that might help.
This lack of organization, the wrong tools, it must kill productivity.
Absolutely. It has a direct, measurable impact. 41% of the survey respondents said data handling caused a backlog in their lab's productivity. Another 22% said somewhat.
So nearly 2/3 are feeling the pain.
Yep. And the problem was even worse. In larger labs, 51% reported a definite backlog. The fields most affected were structural biology, 62%, proteomics, 59%, and genomics, 50% fields generating massive data sets.
And what kind of data causes the most trouble?
Microarrays, those chips used to measure gene expression, and medical images were mentioned most often. Just the sheer size and complexity. And get this, over half of the researchers said they spend more than 5 person-hours per week just on data handling.
5 hours a week. That's like a significant chunk of someone's job just wrestling with files.
Exactly. It's essentially a full day of work lost per person, just managing data instead of analyzing it or doing experiments.
So if they're drowning in data and wasting time, what do they need? What are they asking for?
Well, they anticipate needing much better methods for managing these huge data sets, obviously, and better ways to actually process and analyze the data. They express a real need to move away from everyone doing their own thing, these individual preferences, towards established lab-wide data organization systems.
Like real
The
database.
truly searchable databases, yeah.
Mhm.
Things that aren't hampered by crazy long file names or inconsistent updates or files scattered everywhere.
Okay, the need seems crystal clear. So what's stopping them? Why isn't this happening?
Right. That's the critical question. The study identified three main barriers. No huge surprises here, maybe. First, financial burden, money. 21% felt a specific software product could solve their urgent problems, but the cost is just prohibitive.
Software for labs can be expensive, right?
Very, one researcher put it bluntly. No, whenever I hear the word LIMS, that's Laboratory Information Management System, specialized software, I hear way too much money to deal with. Labs often underestimate these data handling costs when writing grant proposals, and there's a wariness about investing in new solutions, maybe because of past failures or just lack of experience with them.
Okay, cost is number one. What else?
Second, lack of time, just sheer time pressure. The time needed for proper data management, for training people on new technologies, it isn't always seen as an integral part of the research process itself.
It's seen as overhead, not research.
Kind of. Another researcher said it's free in terms of money. It's not free in terms of your time. This leads to what the study called incredibly time-wasteful methods on slow computers by untrained people.
Yeah.
And it's made worse by high employee turnover in labs. New people constantly have to learn these inefficient ad hoc systems from scratch.
Right, the training burden. Okay, cost, time. What's the third barrier?
Third, limited institutional support. More than half of the researchers felt that better support from their university or institution would help immensely. They believe universities, which get indirect costs or overhead from research grants, should provide more basic data infrastructure, things like shared storage, better networks, technical support. That could save individual labs much needed funds.
So institutions could step up more. Okay, that's a pretty stark picture of the struggles researchers face just managing the data, let alone analyzing it. And speaking of analysis, let's pivot to a specific and incredibly fast-moving area, genomics. Historically, the big bottleneck in genomics was just the cost of sequencing the DNA itself, right?
Absolutely. It used to be tens of thousands of dollars for a single sample. Computational analysis, like a few bucks, almost an afterthought in the budget.
But here's the fascinating pivot our sources point out.
Here.
Sequencing costs have just plummeted, nosedived.
Dramatically. We're talking full human genomes for around $600 now. Some companies are even reporting $100 genomes on the horizon.
I'm tens of thousands to maybe $100. That's insane.
It's a complete game changer. And if we connect this to the bigger picture, it means that computational cost and efficiency have suddenly become way more important. They're now a major factor, maybe even the major factor. The analytical pipelines, the software, are now struggling to keep up with the sheer volume of raw sequence data pouring out. We can't just rely on Moore's law making computers faster or Kreider's law making storage cheaper to automatically solve this. The data growth is outstripping those trends.
So what does this flip mean for researchers on the ground?
It means they don't just need to get compute resources anymore. Now they have to explicitly think about and trade off different factors. Accuracy versus speed, compute power versus storage cost versus the complexity of the infrastructure needed.
Trade-offs. Can you
It's
give an
ex-
example?
Sure. Imagine a clinician analyzing a patient's genome for a rare disease. A full best practices analysis might take over 10 hours on a typical server. Or maybe it costs $5 on the cloud, but still takes tens of hours. But if they use specialized hardware acceleration, like something called Illumina Dragon, maybe it takes less than an hour, but it might cost $20 on Amazon Web Services for that speed.
Ah, okay. So faster costs more. A time-money trade-off.
Exactly. And it's not just a technical decision anymore. It becomes part of the experimental design. How fast do I need this result? How much am I willing to pay for that speed? It's a resource allocation choice.
So what are the key trade-offs that developers making these tools and the researchers using them are constantly juggling?
Our sources break them down nicely into technical bottlenecks and then user-facing bottlenecks.
Okay, let's start with the technical ones.
All right. First, compute. It's not just about the theoretical speed of an algorithm, like how fast it scales. Just Parsing these massive files, we're talking 50 to 100 gigabytes, compressed takes a significant amount of time. And analysis pipelines aren't just one step. They're often many small steps, and all those little compute times add up.
Right, it's death by 1,000 cuts, computationally speaking.
Kind of, yeah. Second, space, both storage and memory, RAM. Storage is an ongoing cost. It accumulates. Long-term archiving on magnetic tape might be super cheap, less than a cent per gigabyte. But shorter-term, faster storage on hard drives or SSDs costs more.
And then there's memory. The RAM needed to actively work on the data.
Exactly. RAM is often a hard constraint. If your analysis needs more RAM than the machine has, things grind to a halt or get incredibly slow. Cloud computing helps here, because you can spin up a high RAM virtual machine just when you need it, which is much more flexible than buying physical hardware you might not need all the time.
Okay, compute, space. What else on the technical side?
Communication. Moving the data around.
Right.
This is often a more severe bottleneck than storage, surprisingly.
Moving data.
Yeah. Sending data out of the cloud, for instance, incurs egress costs. Google Cloud charges maybe 12 cents per gigabyte. It adds up fast with huge data sets. And these big cloud-based biobanks, like the UK Biobank or the NIH, all of us project, they keep the data in the cloud, which means researchers basically have to do their computation there in the cloud environment.
Which costs money and requires specific skills, right?
Exactly. It can cost millions for large projects, and you need platform-specific expertise. That limits who can easily access and analyze the data. It's a democratization issue. Another example. For single-cell sequencing, researchers might pre-compute a count matrix to reduce the amount of data they need to move. But doing that means you lose the raw information, which can sometimes skew the results. Another trade-off.
Okay, those are the technical trade-offs happening behind the scenes. What about the user-facing bottlenecks, the things a researcher actually experiences?
Right. These are more tangible. First, time. Simple question. How long do you actually wait for your results?
The turnaround time.
Precisely. Hardware accelerators, GPUs, FPGAs can slash compute time dramatically. But there are non-compute delays, too, like waiting in a queue if you're sharing a high-performance computing cluster. Or just the time it takes to physically transmit terabytes of data or move it between fast and slow storage tiers.
And the time needed depends on the goal, too.
Totally. Research needs like running hundreds of samples in parallel over days or weeks are different from clinical needs, where you might need results for a single patient urgently, maybe in minutes, not hours.
Okay, time. What's next? We mentioned it before.
Money, the explicit time-money trade-off. Pay more for faster machines. Pay for priority access in the cloud queue. If you guess wrong about how much RAM you need, your job might crawl by swapping data to disk, or you might need to stop and request a bigger, more expensive machine. Again, the cloud offers flexibility here compared to buying fixed physical hardware, but that flexibility comes at a per-use cost.
Time, money, what else?
Accuracy. This one's subtle but absolutely critical. Modern methods, especially things like data sketching, sometimes intentionally reduce accuracy to gain speed or save memory.
Wait, they reduce accuracy on purpose? Isn't accuracy the whole point?
Well, it's about sufficient accuracy. Think of the classic BLAST tool for sequence alignment versus the older, slower Smith-Waterman algorithm. BLAST is way faster, but it's technically less sensitive, less accurate in finding all possible alignments. But for many tasks, it's good enough.
Ah, okay. Good enough is the key.
Right. It's about functional equivalence.
Yeah.
Does slightly reducing the precision of an intermediate calculation actually change the final biological conclusion you draw from the whole analysis? Some tools, like one called MASH for comparing genomes quickly, actually make you set parameters that directly control this accuracy-speed trade-off.
So how do you know how much accuracy you can sacrifice?
It's tricky. A good rule of thumb might be you can reduce accuracy up to the point where the uncertainty it introduces is roughly similar to the uncertainty you'd get just by rerunning the original biological experiment.
Okay, that makes sense. Time, money, accuracy.
Yeah.
Is there one more user bottleneck?
Yes. Complexity. Just the sheer difficulty of developing, deploying, and using this high-performance software. Developers often have to use low-level languages like C++ for maximum speed. Then they might wrap that code or glue it together using easier languages like Python.
That sounds complicated.
It is, and sometimes that glue code itself can take a surprising amount of runtime, especially if you're dealing with messy or non-standardized file formats, which is common in new, rapidly evolving fields like single-cell analysis. Plus, just setting up and configuring high-performance bioinformatics software often requires specialized skills. It's not always plug and play.
So it's a challenging landscape with all these trade-offs. But are solutions emerging? Are things getting better?
Oh, definitely. There are some really clever approaches being developed to tackle these bottlenecks head on.
Like,
Well, data compression is a big one. Classic compression saves storage space, but you often pay a penalty in compute time to decompress the data before you can use it. So now there's a push towards compressive genomics techniques that try to operate directly on the compressed data, like in standard formats called CRAM or BAM, avoiding that decompression overhead.
operate on compressed data.
Cool. Yeah. And there's also lossy compression in genomics.
Aussie, like JPEGs for images where you lose some detail.
Exactly like that.
Yep.
Discarding information that might not be critical for downstream analysis, like maybe detailed quality scores for every single DNA base to achieve much higher compression ratios. Again, it's that accuracy trade-off, but applied to file size.
Interesting. What else?
Data sketching. This goes even further than lossy compression. Instead of just compressing, it extracts only the most important salient features from the data, creating a much smaller sketch.
Like a summary of the data.
Kind of. It allows for incredibly fast comparisons and analysis because you're working with the small sketch, not the massive original file. Examples include MASH, which we mentioned for estimating genomic similarity very quickly, or minimizer-based approaches, like the popular tool Minimap2, for rapidly mapping sequence reads. For single-cell analysis, there are even geometric sketching methods like GeoSketch and Hopper that can create representative sketches of cell populations, sometimes even improving the detection of rare cell types compared to analyzing all the data.
So sketching can actually improve results sometimes while being faster.
In certain cases, yeah, by focusing on the essential patterns. Then, of course, there are hardware accelerators.
GPUs and FPGAs.
Right. Graphics processing units and field programmable gate arrays. They're being used in commercial platforms like Illumina's Dragon or NVIDIA's Clara Parabricks to provide massive speedups for specific common bioinformatics workflows, like aligning sequences or calling variants. They require specific programming expertise and investment, but the speed gains can be enormous.
Okay, compression, sketching, hardware. But what about the cloud?
Cloud parallelization is huge. The cloud gives you on-demand access to a vast range of different server types, solving that problem of needing specific, expensive hardware locally. It also helps centralize data storage and compute for these massive biobanks we talked about, reducing overall costs and enabling huge reanalysis projects across thousands or millions of samples that would be practically impossible otherwise.
And finally, making the software itself easier to build.
Yes, that's the goal of domain-specific libraries and languages, trying to simplify bioinformatics software development. Libraries like BioPython or RustBio provide reusable building blocks for common computational biology tasks, and domain-specific languages like SnakeMake or the Galaxy platform make it much easier to define and run complex multi-step analysis workflows without writing tons of custom code. There are even newer programming languages being developed, like one called Sec, that aim to combine the ease of use of something like Python with the performance of compiled languages like C++. That's kind of the holy grail.
So lots of innovation happening to try and tame this data deluge and manage the trade-offs. It really feels like the world of biomedical data is, well, a classic double-edged sword.
It absolutely is. On one side, you have this immense, almost unbelievable promise for scientific discovery, for personalized medicine, all fueled by sequencing getting cheaper and cheaper.
But on the other side,
On the other, you have these profound challenges around privacy, the practicalities of data management we just discussed, and the very real and growing costs of computation.
and what's really striking from our discussion is how intertwined all these issues are, isn't it?
Completely. The need for faster analysis drives new tools, which create new trade-offs. Privacy concerns shape how and where data can even be shared or processed in the first place. It's this constant dynamic balancing act between getting the most utility from the data and ensuring adequate protection.
And as the amount of data keeps growing, the complexity increases, more of it gets generated in real time, maybe from your smartwatch, your fitness tracker.
Yeah,
The decisions about how we manage, analyze, and protect it are only going to become more critical.
And it's not just an issue for the experts in the lab or the cloud architects. These decisions directly impact what research actually gets done, what breakthroughs are possible, and ultimately what might be possible for your own health down the line.
This whole deep dive really makes me wonder. As our ability to generate and analyze more and more of our own biological data increases personally through consumer tests, wearables, whatever comes next, how much control do you, the listener, truly want over its use? And what trade-offs are you personally willing to make between your privacy and the potential benefits of scientific advancement for everyone? It's something to think about. That's it for this deep dive. Join us next time for another journey into the fascinating world of knowledge.